Good day, everybody. My name is Martin Beukema, and on behalf of the next generation of vaccinologists of the ISV, I welcome you to our monthly webinar series. So the next generation of vaccinologists of the ISV are dedicated to enhancing the visibilities of early career researchers within the field of vaccinologists, for example, through the organizations of those webinars. So if you would be interested in giving a presentation during one of our webinars, or being interested in joining the organizing committee, please email us through the email addresses you can see here on the slide, or find us on the ISV website. So today we will have two exciting talks and presentations from Danilo, Casimiro, together with Mona and Chile, and together with Alper Sevigel. But before we continue with the presentation, I would like to draw your attention to a few other points. So first, I would like to draw your attention to the annual congress that will this year be organized in October in Seoul. So I hope you would also be able to join this conference. But if not, you would also, if you're not able to attend in person, there's also a virtual platform where you could join, through which you could join the conference. Then I would also like to announce the next webinar organized by the ISV on the 25th of September. And so during this webinar, Laura Palomares from the National Autonomous University of Mexico will give a presentation on the immunogen structure and production method as determinants for vaccine quality. We also hope to see you all there. And lastly, I would also like to point out actually the benefits of being an ISV member, and especially for early career researchers. So only for $35 US dollars, you would have a 1-year membership, and it gives a lot of benefits for you as an early career researcher. So for example, you could join the mentorship program that has also recently been established and is dedicated of providing guidance for professional development of trainees in vaccinology. Please find the activities of us or the ISV through this link of QR code. You can scan it now, for example, and it will guide you through our LinkedIn. But you could also a lot of activities of the society on the website. And at last, I would like to announce our next webinar as well from the next generation vaccinology webinar series, which will in which Celine Lemoine and Gerrit Borgaert will give a presentation on the 10th of October on 4 o'clock Central European time.
Martin?
Yes.
I don't think your slides were shared.
Oh, really? Oh.
I'm sorry.
So, this is really a pity. So then I will quickly go. So this was the presentation from Laura. Please join us through the membership, and here's the QR code. So I hope you can join us through the QR code. And after that, I would like to stress again the presentation on the 10th of October for our next webinars. With that, I would like to give the word back to Michael, who will be chairing this session.
Okay, thank you, Martin, and welcome everybody. Thank you for joining us today. So this is going to be a busy webinar, a very interesting webinar. We're going to have two presentations basically. The first one will be on career development, and with some very interesting talks from colleagues, vaccinologists from Sanofi. The second part will be scientific on biomarkers and predictive, and how they can be predictive for vaccine responsiveness. So the first talk will be given by three speakers, which is Dr. Danilo Casimiro, Dr. Qinglin Xie, and Dr. Mona Motwani. Dr. Casimiro is the Chief Scientific Officer and Global Head External Scientific Affairs at Sanofi. Dr. Xie is a structural biologist with a PhD from Cornell, now working at Sanofi. And Dr. Motwani is Principal Scientist and Immunologist with a PhD from UMass. And so we're going to start with the career panel. We're looking forward to your presentation, please.
Excellent. Let me just put this one on presentation mode.
Yeah,
Can you see it?
this is perfect. Thank you.
Okay, excellent. Well, thank you everyone for the thank you ISB for the opportunity to speak with the early career researchers and scientists of the ISV. And we will do a tag team here with Mona and Ching Lin, two of our early career hires within Sanofi. And I hope they can share with you and motivate you in terms of joining the biopharmaceutical industry. Now, I've structured the talk in a number of ways and in the following manner, and that is I'll share with you the entry point most commonly used to get into the biopharmaceutical industry. I'll share with you as well what one typically see in the environment within the biopharmaceutical industry. I'll share a little bit about Sanofi vaccines, which is really representative of many of the biopharma vaccine companies that exist today. And a little bit about the opportunities within the biopharmaceutical industry, specifically in our vaccine R&D. Then I'll move on to Mona and Ching Lin. So this slide shows you the diversity of professions in the biopharmaceutical industry, ranging from research and development, services, commercial operations, and operations, specifically manufacturing in this case. But for scientists, the most common entry point is the R&D. And in fact, in a large pharmaceutical company, many of these services exist within the organization. However, with smaller biotechs, they focus more on their core competency, and many of the other services or professions are outsourced or within an external network. And most of the time with biotech companies, the core competency is in the R&D space, right, that they keep internally. So it shows you here, what I want to show you here is that typically the entry point for scientists would be in the R&D space. But as your career progresses, there are certainly plenty of opportunities to move to other professions within the biopharmaceutical industry. And in some cases, it requires additional training or additional degrees. For example, if you want to go into legal, we have scientists who are lawyers who used to be scientists as well. So it just tells you the wealth of opportunity here in the industry itself. What does it mean to be in the biopharmaceutical industry? I mean, it means many things. Creating breakthrough medicines and vaccines. That's the most important. That's primary in terms of our mission. We challenge you in terms of working on challenging science or hard science. We always embrace new ideas because we have to evolve as we go along. We have to embrace new science and new technologies. And you're expected to develop the expertise to build skills really for tomorrow. And what's important here is that you will be part of a team. And that teamwork is very important in terms of being within the industry here. Because as you can see, there are many players that it takes a village to develop a drug or vaccine. Now, I just want to stress here that within pharmaceutical companies like Sanofi, Merck, GSK, there are multiple therapeutic areas. And it's not uncommon that one would join one therapeutic area, but could move to other therapeutic area, especially if your expertise or your background is fairly transversal across the different areas of focus. For example, in Sanofi, we have specialty care, specifically on immunology, inflammation, rare diseases, blood disorders, neurology. Then you have on the far right, vaccines, and general medicines between specialty care and vaccines. Now, to give you an idea of what Sanofi vaccine is like, this summarizes who we are and what we do. We have 5 million people vaccinated annually with our vaccines. We have 2.5 million vaccine doses supplied every day. And our scope expands to about 150 countries. In the case of polio vaccine, we have 395 million doses of vaccine supplied to UNICEF since 2014. What I'd like to highlight here is that we have a global footprint. We have 6 vaccine R&D sites from North America, the US, and Canada, Europe, specifically in France, and also in Asia Pacific. We invest about €1 billion in vaccines R&D every year. And as many of you know, we are a world leader in influenza and pediatric vaccines. Now, when you come into the biopharmaceutical industry, we select, definitely you're very strong in the science, right? You're very familiar, you're very strong in executing or discovering subunit recombinant vaccines, monoclonal antibodies. The skill set are there. But one thing is important when you join a biopharmaceutical industry or company is that it's the ability to transform what we know in science to actual candidates and products, right? This arrow right here is the most important part of what it takes to be successful in the biopharmaceutical industry. You have a strong science on the left, but really in the end is translating that science to products and candidates that will go on clinical development. And in case of Sanofi vaccines, these are many of the portfolio candidates that we have currently at hand. Success, obviously, in this case, is only ensured by the excellent talent that's hired across the entire R&D chain, going from vaccine and antigen design, immunology, CMC, whether it's mRNA or non-mRNA, all the way to clinical and regulatory. And I have here not an exhaustive set of scientists or scientific expertise that could qualify for each one. I'd like to point out, though, that there's also this strong emergence of digital machine learning and artificial intelligence that's now transversely applied across the entire value chain. So it doesn't mean you have to train in digital AI, ML exclusively, but as you can see, many of biologists, structural biologists, protein chemists would seek it as part of their training, being able to apply and even develop AI and machine learning tools. And I'll now give it to Mona and Qinglin, who'll speak about their journey, as well as why they joined the biopharmaceutical industry and how their aspiration of growing within the biopharmaceutical industry. Let's start off with Mona.
Hello, everyone. Thank you, Danny, for the introduction. So my name is Mona Motwani. I'm a principal scientist at the mRNA Center of Excellence at Sanofi. So my motivation to join industry was really this early exposure to working with industry partners during my PhD. So I was working on a particular molecule that was a very attractive drug target. So we had extensive collaboration with companies like Merck and Biogen. And it was very interesting that both the industry side and the academic side were looking into the same basic research questions, but our conversations around industry partners were more in line of translational value of the work we were doing, the clinical relevance of the work or the data we were generating, how this data would help a patient in the clinic. So I thoroughly enjoyed that, and I decided right after my PhD that I want to be in biopharma industry space to really see the transition from bench to bedside. And since joining the industry, my experience has been very satisfactory and empowering that the work we are doing today will help support a drug product that will really impact people's lives. So that's what my motivation came from to join industry. So I joined as right out of my graduation as senior scientist at Sanofi at the Genomic Medicine Unit, where we were focused on rare diseases using AAVs or viral vectors as gene therapy to cure these diseases. And my expertise as an immunologist was utilized because we were looking at immune responses to these AAV vectors. And Sanofi or any industry really provides this very collaborative environment where you can really showcase your skill sets and apply it to different variety of therapeutic products and areas. So I really took advantage of our internal cross-functional collaborations, team meetings, webinars, seminars to really showcase the skill and data that I had generated. And I was hired by the mRNA Center. So I still do what my basic technique or expertise is to look at immune responses, but now we look at it differently where I'm looking at mRNA LNP vaccines. How do they induce immune responses? And really trying to balance efficacy and safety to develop better vaccines. So what's beautiful about industry, or what's also exciting, is that it's very diverse, but it's very cross-functional. So you still do what you love, but you can apply it in a lot of different areas. And similarly, this also provides you an ecosystem where you can grow as a scientist or also take a path to grow as a manager. So my recommendation to young researchers out there would be to really showcase your work, showcase your skill sets, your talents internally, externally, and really seize every opportunity to make connections or establish your networks, because this will really help you develop and grow in your career. And now I'll hand over to my colleague Jingling. Hope my experience helps others. Thank you.
Thank you,
Okay,
Mona.
thank you, Mona. So yeah, so I'm Chin-Ling. So could you move the slide to the next one? Thank you.
Okay.
Yeah. So I'm Chin-Ling Shea. I'm currently a senior principal scientist in vaccine antigen design. So when I joined the bio industry is actually a very different story. So back to my postdoc training, we were using structural tools such as cryo-EM and X-ray crystallography to get in high-resolution structure of the viral antigen. Then we're using the antigen structural information as a blueprint to design a more stable protein for vaccine. So wonderful example actually is the COVID-19 vaccine and also RSV fusion protein vaccine, which has been very successful licensed by Big Pharma. And then I was very lucky to work with wonderful project with Sanofi, and then Danny was there as well. So where I start to see different things between pharma and the in academia. So back to the lab, we only focus on antigen design. So, but there are way more information you can extract from the human immunology. So once you immunize the vaccine candidate to the human, and then we can sort of like collect the infected like vaccinee, PBNC, or infect human subject, and start to probe in their PBNC, their antibody, how they respond to the vaccine antigen or the infectious disease. Yeah, and then I guess after I joined the company, I start to growing from an individual contributor to become sort of like part-time manager role. So I was able to more like delegate to more project to different people, and then doing more cross-functional collaboration with different unit within Sanofi, and even sort of like reinventing what we think about the vaccine. So vaccine not just for infectious disease, it could be for maybe neurodegenerative disease, and even using your imagination, maybe vaccine can against aging. It's also possibility as well. Yeah, and then in Sanofi, we were also able to collaborate with academia and the government agency. So as you guys might know, Sanofi is very famous for flu vaccine. So there's a lot of opportunity you can talk to like NIH or broader like FDA government agency, and to help them to design a potential vaccine to prevent a future pandemic. So thinking about like your design can make it from your own hand and then go through to the human clinical trial and and solve and potentially prevent the pandemic happening. I think it's pretty cool. Yeah, so I guess the main reason I joined the company is because you can really see your own design from beginning to the end. Goes through the quality control, manufacturing, and even to a clinical trial. And once you got a result from clinical trial, then you can use those information, especially human immunology data, to inform your next generation design. That's pretty much it.
Okay, thank you very much for this very interesting presentation. And so we have now opened this presentation or these presentations for questions. You can either raise your hand and then we will unmute. And you can ask your question yourself, or you can drop your question in the Q&A box. And maybe I can kick off with the first question while people put their questions in the Q&A box. So thanks again for this very interesting experiences that you share with us. And a first question that I have, and that's people ask very often from academia, is there an ideal time point to move from an academic setting into a company? For example, you have taken your experience into the company, it seems. So, but do you have to do, for example, a postdoc, or can you immediately after graduating move into a company for R&D work?
Yeah, a very good question. I hope you can hear me, right, Michael?
Yes.
There's really no optimum time. It's really the opportunity presenting itself to the person as well as for us as well. When we hire or put up a job description out there for an opening, we would either say no experience after a PhD, which could be 0 to 5, or in some cases we would require 5 to 10, or 2 to 10, which may be including a postdoc. So it depends. There are multiple levels of entry into a company. There are really straight PhD hires, which would be certain level, different level. They're called differently in different companies, right? I came in after my postdoc, but more recently there are really examples where they're coming in directly from their PhD. And especially if you have a competitive market for certain skill sets that in this case, you could hire as early as a PhD graduate. But it depends on the opportunity that's being presented externally, as well as the required skill set we have. But it varies. There's no strict rule in this case.
I see. So
one thing I'd like to point out, though, is that if Mona, which we've talked about with Mona as well as Ching Lin, is that there are also opportunities for industrial postdoc that will provide you that exposure early on, immediately after your PhD. Industrial postdocs are offered by many pharmaceutical, even biotech companies, that you can get that early exposure. In fact, that sometimes could be more equally valuable, if not more valuable, for someone who's hiring, because you have the exposure in the company as well. And in some cases too, we have at Sanofi where it's not a postdoc, but we sponsor PhD students for 1 year in the lab as part of their doctoral training.
I see, very interesting. Okay, so
Ching Lin or Mona, do you want to add?
I also wanted to mention about co-op opportunities. So basically, we also have co-op opportunities where people, at least in my experience, join immediately after their master's. So there's not really like an age group criteria. It's really the opportunity and if you are a good fit for that opportunity. So you've, like Danny mentioned, postdoc opportunities, co-op opportunities, sponsored PhDs, if you can grab those opportunities to really enter industry at any stage and age in your career life.
Yeah, I just want to second Mona. We do have a co-op position opening at the end of the winter, the beginning of spring in structural biology. So people who has a PhD in structural biology, please apply. So you get to test about like how industry work. Yeah, I think it's pretty cool. Even in cryo-EM or X-ray crystallography, we work differently from compared to academia.
And that helps you build your resume, right?
Yes, exactly. Yeah, and you get to know people, and then people know you are a good team player. Team player is also very important quality in industry as well.
Yeah, so that so we start to have some questions in the Q&A box, and some of them are more or less already answered. But the first question from Annika Luo was, in your experience, what was the biggest learning curve or difference between research and academia and industry?
It's probably more fresh for Qinglin and Mona. So why don't I give them the opportunity to answer this one?
I can go ahead. So I guess the biggest learning curve here was like in your PhD, you're really on your own. You know, you have an experiment today, you'll do it, maybe take a week and then come back and go and show it to your mentor. But industry is really more focused. It's more focused work you do so that you don't wander off, you know, because so it's more focused work, more accountability also. So we work in a framework of goals and what is our objectives and what experiments are we really doing to meet our goals. So I guess the learning curve, which was good in a way, is it really makes you more focused, oriented, goal oriented, and working together, because your result will provide knowledge to someone else so that they can begin their work. So it's very much like an assembly line where you have to be really good at your job, give them enough starting point so then they could take it off to next level. And that's how we go from research to development to, you know, safety to manufacturing to really into the clinic. So for me, that was the learning curve, transitioning from academia to industry.
I guess for me, it's developing my soft skill and then communicating with different team members among multiple different units. So in academia, you work in a lab and then you report to your PI, and then you probably have more freedom of operation. But in industry, you need to align with multiple teams. So like there's a team doing antigen design. Also, there's a team doing evaluate immunogenicity in mouse study, and also thinking about manufacturing and also epidemiology. So it does really need to align, because eventually you want to bring a vaccine to be your candidate to become a real vaccine product, right? So you also need to fit the business needs as well, right? So I would say like in industry, you might have a chance to join a global project team where you can talk and communicate and align with different experts from different fields.
Okay, thank you. So we have time for one more question. I suggest that if we move on to the next presentation, maybe the colleagues from Sanofi can answer the questions that remain in the chat box. But one more question. So thank you for your very interesting presentation. Please share the best tips for getting a job in industry. Is it okay to just apply, or do you need to have a contact in the company?
That's I don't think there's one fixed set of rules in this case. It does help that you have that it does help sometimes that there is historical appreciation of the science. I think when I was hired, I published quite a bit. So you can see that one thing I could share is the following. This is related to the first question, is that when I was hired at Merck, there's one thing that resonated with me that they said: we hired you because you can move from one project to another project with success, right? You're not just type 1 diabetes person, because what's important in this case is that you have the talent. You can show that you have the talent, the agility to move from one project to the next. And sometimes that shows in your CV, right? The diversity of work that you do. So that shows in your CV. So CV, preparing your CV is very important because that's the first entry point. I mean, you can imagine when we open a position, there will be 300, 400 applicants. We can't go to 300, 400 applicants manually. There are now AI tools, screening tools that will narrow that one to a few applicants that we will look at. So the CV is a very important aspect of what you have to pay attention to. Successful CV writing is important, but obviously it helps also to have some network. As you build your network in terms of, hey, if you're a professor who has contacts with biotech companies, they can help you, recommend you for when a position opens up. As I mentioned to Mona, my first position, I didn't know anyone. I got in on the merit of what I've done for my PhD and postdoc. But my succeeding positions, it was really because of people who I worked with in the past that got me through the gate. So combination of many things.
Thank you. So Mona, Chinglin, do you have something to add to this question? Because we have to move on to the next talk, but I suggest that for the remaining questions, maybe you can put your answers in the chat box if you don't mind. Some of them are relatively specific, but it will be highly appreciated if you could do that. So with this, we're actually moving to the next talk. So this was, thank you very much. It was amazing. And now we're going to move from the career panel to the scientific presentation by Alper Severagel. I apologize.
Hello, everyone. So I hope my screen is sharing.
Can you swap the screen?
Yes.
Swap the display. Yes.
Okay, perfect. Hello, everyone. It's a pleasure to be here. So today, in the second half of the webinar, I will share my PhD work. So I'm currently finishing my PhD in the National Institute of Public Health and Environment in the Netherlands. And today I will talk about understanding the pre-vaccination immune landscape to identify biomarkers of vaccine responsiveness. So I would like to start with giving an overview of aging and why we are interested in aging and vaccine response, and what happens to our immune systems as we age, the clinical trial we were performing, and how we identify these biomarkers that we are interested. So to start with, we are interested in aging because our population is at the moment very rapidly aging, especially in Europe. So this red line here represents the ratio of older individuals to younger individuals in the population, and you can see that it's rising quite steeply. And this leads to some problems. For example, older population is at higher risk of infectious diseases, but also there's more disease burden in the population. This increases the healthcare costs, and also lack of personnel to take care of this increasing demand. So these are becoming an evident problem for our near future. So that's the reason we are interested in studying aging. And what happens with aging when we talk about like what really aging means, it's actually just chronologically growing older. But we are interested in from a biological point of view, where we study progressive and gradual decline in the biological functions. And aging is a very complicated process. It can be functional adaptive modifications that happens during aging, or they can be dysfunctional and maladaptive. And the real problem that we are facing with the immune system and the aging is the observed immune dysfunction that associates with chronological aging, and this is called immunosenescence. And as we age and as the immunosenescence kicks in, we see decreased immune surveillance in the tissues, in the organism. The response to pathogens are getting weaker, and what we are interested is especially is response to vaccines are also getting lower. So the aims of my PhD project was to understand age-related changes in the immune system and how to study these. and with the goal of identifying individuals who are at risk of lower vaccine responsiveness before we give these individuals the vaccines. Because the next challenge in our healthcare and public health will be targeting guided personalized vaccination strategies for these people to ensure their protection and decrease the disease burden in the population. So to study this, we had preclinical cohorts of younger to older individuals. So these are 25 to 98 years old, around 300 people. And these people received 3 different vaccines over the course of years. And from different time points, which I will not mention all of them, but I will mostly mention the pre-vaccination start time points in this presentation. We collected blood samples, and using these blood samples, we investigated how the immune system of these participants behave and how does it look and what can learn from it. To study, of course, the age-related changes and what happens to the immune system before after vaccination. And to study this in this cohort, we use flow cytometry mainly that I will discuss today to get an overview of T-cell, B-cell, NK cell, monocyte, and granulocytes. And to study vaccine response, to study the functionality of these people's immune system, we use vaccine response as an output. And then we did this with antibody measurements, antibody assays, and so forth. So I think the important thing here is, in the essence, we want to understand who are in this cohort that have weak immune function and who will not respond to vaccines. So what we really need to understand is how we approach vaccine response and immune function. Because vaccine response is a very complicated output. You can define it in many different ways. It involves too many factors. And immune function at the moment, where we are now, it's not very easy to say this person has a good immune system or bad immune system. So to study immune function, I think we propose that the good idea is to approach it as a high-level emergent property of the immune system. And these emergent properties are present in many complex systems. So basically, the essence is complex system exhibit these properties, but these properties not always directly related to properties of the constituents of the system. So I will give a very simple example. So if you look at a snowflake, the constituents is actually a water molecule. But if you study a single water molecule, we will never understand the complexity of a snowflake, because there's a lot of other high-level emergent complexes are forming with some simple ingredients. So this is similar in biological organism, yet much more complicated. But in this case, a single immune subset or single biological measurements in general will not tell us how the immune system of a person will behave. So what we need to do is to understand the total snapshots of the variables we have at the same time to define the pre-vaccination state of these individuals. Because if you look at the single variables, such as naive CD4 T cells, which are one of the most important age-related changes reported in humans, we see a steep decline in older individuals, but more importantly, the heterogeneity in the population is also increasing as we get older. So one of the key factors to understand this picture is, we believe, is related to this heterogeneity and understanding where this heterogeneity is coming from and how we can study this complexity to really find out people with weak immune systems. And this heterogeneity is also very important because this is one of the reasons that one solution will not fit everyone in the population, because older individuals are much more different from each other than younger individuals. And this really pushes forward the idea of personalized vaccination and medication strategies. So to understand this heterogeneity, I will very briefly describe a landscape of immune variables. So based on all the immune variables we can think of, we can identify a person in this landscape in a certain location. And our immune systems in this landscape, our immune system in this landscape is shaped by different variables. And one of the most important ones are, of course, age and environment. Because our immune systems are constantly under pressure of environmental factors, pollution, or diet, or pathogens, chronic inflammation, or chronic infections. And all these factors are pushing our immune system in different configurations in this landscape. So what we see in older population is through this remodeling and adaptation and aging, we observe these complicated, different, heterogeneous phenotypes. And of course, the idea is that some of these individuals age healthy, so they maintain their immune function and respond to vaccines and don't develop pathologies. But some others develop immunosenescence and show weak responses to vaccination. So to study this complexity, and associated with vaccine responsiveness, we propose the following. So this heterogeneity that is increasing in the older population should be related with the vaccine responsiveness that we observe in the older population. So this basically the variation in the immune system should have some signatures that we can use to study vaccine responsiveness at the pre-vaccination time points. So then the key to understand the vaccine response in this case lies in the understanding the heterogeneity of the immune system at the baseline before vaccination. So to study this heterogeneity in the population, we did one of the most simplest thing you can imagine is basically categorizing these people that look alike based on their immune systems. And the reason to do that is many systems, physical or biological systems, they contain baseline properties. And these baseline properties can tell what the system will do when it's stimulated or when it's challenged. So if we categorize people with similar baseline states, then we would expect them to respond to vaccination in a similar manner too. So to get these categories, we basically used all the baseline immune subsets we measured. We simply clustered them, and we identified for this cohort 9 different groups of individuals, which we call immunotypes. And then we investigated the differences between age in these groups. And you can see that there are 65 plus individuals that show different phenotypes. And there are some younger individuals are more restricted in some other immunotypes. And of course, together with age, cytomegalovirus infection was also very prominently different between these groups, showing that the variation, these 2 variables are some of the most known associates of immune variation in the population that we can also see in these immunotypes. So basically, instead of using immune system by subsets or variable by variable, we grouped it together to study to respond to all these vaccines. So basically, what I'm going to show you here is using these 9 groups of individuals that with similar immune system configurations before the start of the trial, and then vaccinating these people with influenza, pneumococcal, and corona vaccines, and collecting antibody serum and plasma to measure antibody responses, and to basically study this vaccination response. What we did to combine all these vaccines together as a one score for multiple vaccine responsiveness, we use a quartile approach. So basically, for every vaccine, individuals are split into 4 quartiles, and people in quartile 4 then would be in the highest antibody titers or functional outputs compared to people in quartile 1, which are the lowest responders. So basically, if you do this for every vaccine, and here I show these in columns, for influenza, pneumococcal, and corona vaccine, and these quartiles represent the response categories for individuals. And if you follow individuals across different vaccines, you can see that some individuals, a lot of individuals move in between vaccinations. So this is also interesting because the fact that one person responds to one vaccine strongly doesn't mean that they will do the same for another. because the mode of action is different, pre-existing immunity is different, etc. Which we corrected with some models, but I will not go into detail. We can discuss later. So basically what I want to highlight here is, although there is a lot of movements in response between this vaccination, there are still a group of individuals that are constantly at the lower ends of the response. And to simplify these 3 different vaccines and 3 response categories, what we can also do, just take an average response category for every individual, which gives us this simple score of your overall responsiveness to these 3 vaccines. And then we can investigate how the immune phenotypes that we identified at the baseline is associated with response to this 3 vaccine average. And here you can see that in average response quartile that are the highest responders, the blue immunotype, this is immunotype 1, has the highest proportion, and this is decreasing as the vaccine response is decreasing. On the other hand, this immunotype 8 has the highest proportion of lowest responders, and this is also not even present in the highest responding individuals. So basically, to test this hypothesis, we formed an orbital logistic regression model. So this model looks at the difference changing quartiles for the average vaccine response. And then we investigated how immunotypes are associated with this average vaccine response. And here we can see that individuals who showed immunotype 8 were indeed significantly lower responders, whereas immunotype 6 and immunotype 1 individuals showed significantly higher responses. And if you look at the characteristics of these immunotypes, we can see that immunotype 1 are the most youngest group in our cohort. So these are basically our healthy young controls that are median age 41 years old, and their immune system shows all like youthful associated immune signatures. You can imagine like high naive response, high naive T cells, lower myeloid cells, etc. And if you look at other high responder group, these are the older adults with median age 66 years old, but still responding quite strongly to vaccines. So these, we can say that these are healthy aging adults that kept their immune system function intact at older age. And if you look at immunotype 8, and these correspond to low responder older adults with median age 65. And in these individuals, we see very striking age-related differences in the immune system, like very low ratios of naive to memory cells. Also, numbers of these cells are extremely low B cell numbers, and quite high increased follicular T cell signatures. Of course, we cannot say that this is the reason why these people don't respond to these vaccines, because we are just characterizing what we measured based on these individuals, and these age-related changes not necessarily explain the vaccine response itself. So we need something better to summarize this information about immune aging and associated with vaccine response, because categorization is also often not the best approach, as the categories are highly dependent on number of people in the studies, types of variable measured, etc. So what really makes these individuals high or low responders? That's the next question we went after. And to do this, I will explain a very simple metaphor using just 2 cars. So if you let's say you're gonna do a very long trip in the United States for weeks, and you're gonna rent a car, and then the question is, you want to rent a car based on how old the car, because you want to make sure that you're not going to be having a breakdown in the middle of somewhere. So of course the risk factor for systems to malfunction is really age. Age is a very important factor, because with age systems get more dysfunctional. But if you look at how much this system was experienced to exposures, environments, or how much it was used, then you may even expect that a car that is younger but in a worse shape could be a worse choice for your goal. So we think immune system works in similar way. So some individuals, despite being older, they keep high function because their immune system is in high homeostasis. However, other individuals, they accumulate wear and tear at high rates, and their immune system gets compromised. And what we really need to associate immune response to immune system characteristics is measuring this difference between individuals in our cohorts. So to do that, what we did was a distance calculation, actually. And this distance calculation is what we call immune entropy. So this works as setting a young reference group, because we know young individuals have high immune response, and their immune systems are very similar to each other as well. So using this young reference group, we basically calculate distances using all the immunological variables we measure before vaccination, and we calculate a distance. So our hypothesis is that if the distance is low, regardless of age, these individuals kept their integrity of immune network, and then they would respond to vaccinations strongly. But if the immune distance is very large, similar to the car that has been driven like 400,000 kilometers, then there's a higher risk of immune malfunction in these individuals. So we basically calculated this score for everyone in the cohort. And then first we plotted it for each immunotype. And if you look at the score, the high responding immunotypes, which were 1 and 6, they are the lowest ones in our cohort, whereas the low responder immunotype 8 shows the highest significantly different immune entropy. Or if you look at the average vaccine response, plotting the immune entropy, you can also see that the individuals with the lowest response quartiles, they have increasing immune entropy as well. But more importantly, we need a variable that works regardless of age, and that's we can disentangle the age components. So if you make a model and includes age, CMV, and sex, which are important variables that shape immune variation, immune entropy is still significantly associated with multiple vaccine response, suggesting that this sort of calculation to measure immunological distance could be associated with vaccine response across the different vaccines. We also did it for single vaccines, but for the sake of time, I'm not going to go into details of that. And of course, this is one cohort and in one specific setting. So we used another cohort that we had for the Pfizer vaccination this time, 67 individuals, and for these individuals, same immunological variables were measured. So if you look at these individuals, we see a weak association between immune entropy and antibody response to Pfizer vaccination too. And then there were left some people that did not receive the Moderna vaccination, but did receive Pfizer vaccination through the National Vaccination Program. And if you include these people as well in the cohort, which were not included in the previous analysis, then we see that this association is even getting stronger. So there's evidence that this works for different number of people as well. And another interesting thing is we also have serum measurements in these individuals, and one of the important thing is for the serum proteins is a phenomenon called inflammaging. So the high low grade inflammation in the blood is associating and driving immune variation and immunosenescence, as people believe today. So if you look our immune entropy measure and if you make simple models for the serum markers after including age, sex, and CMV, we still see 3 key cytokines that are and serum proteins that are significantly associated with this variable, highlighting that this could be also interesting to study, not just by using immune cell subset information, but also serum markers or other types of data as well. And lastly, we also wanted to see how about a disease cohorts, because there are patient cohorts that's significantly low responders to vaccines, and some of these are kidney transplant recipients. So kidney transplant recipients are, of course, under immune regulatory medicine, but also in general very low responders to vaccine. There is also immunological components, and it's still not clearly understood what's going on. So we received data from kidney transplant recipients who received Moderna vaccination, and we calculated immune entropies for these people too, using an age-matched group. And here we can see that these individuals had very significantly higher immune entropy scores as well. So this could suggest that this also could be relevant in disease setting too. So lastly, what we think is happening, why this sort of measurement is working. With aging, there's a lot of structural changes in the tissues. And one example is lymphoid tissue. And cells cannot be in the right place in the right time. And this weaker cell communication with suboptimal survival signals, they affect the composition of immune cell subsets in the blood. And what we measure by this composition is associating with the detrimental effects of aging on immune function. So with that, I would like to conclude to have some more questions, so have some time for questions. So what I wanted to show you is using this categorical approach is a good way to study heterogeneous immunological states in the aging population, because they can describe age and CMV-related alterations and also associate with vaccine response even, highlighting that the understanding the baseline can really tell us what the system will do upon vaccination. And we can go further and summarize what we see in immunotypes in one single variable in the form of immune entropy. And here we see the extreme immune variation is actually what's causing detrimental effects of immune aging. And this is significantly different between vaccine responder groups. And there is also a lot of potential to improve this. So we measured external variables that we think that will be increase the resolution of this measure. And we see that there are novel naive T cell subsets very strongly associated with entropy, but not as strongly with age. And these are also very significantly associated with vaccine response. So incorporating key elements of immune aging into this equation could help us even understand this relationship much better. And of course, using healthy and diseased cohorts together with multi-omics approaches and applying this sort of thinking can hopefully help us to figure out and define immune systems that are at risk of pathologies or not responding to vaccines. So with that, I would like to thank young next generation vaccinologists for giving me the opportunity to talk about my projects, and also my supervisors, Debbie and Anna-Marie. And with that, I'd like to conclude so we can have questions. Thank you.
Thank you very much, Alper. So this presentation is open for questions. And maybe I can start off. We have time, not too much time for questions, but maybe since I don't see any in the Q&A box so far, I can start with a question about your very interesting approach. Do you think it's possible to come up with new correlates of protection? So new correlates that would predict if a person from a certain immune type that you define is better or worse protected against infection or severe disease during infection, for example?
Yeah, so I think using something like this can help studying different correlates of protections indeed, because what we also see, there is vaccine-dependent response. So some people are so much better responding to viral components than bacterial components. So basically, using this sort of categories, you can push that forward indeed to study a different course of protection.
And have you ever applied this approach on adjuvanted vaccines? Because I can imagine that an adjuvant would change your immune type or your entropy at least.
No, so we only have these 3 vaccines at the moment, but Yeah, that would improve the response, right? But even with high responder settings, for example, with corona vaccination, people respond very robust. But then we still see among those people with immune signatures that relate to aging, they're at the lower end of protection. So even if you maximize the vaccine response and say that people are protected, then there's antibody decay and there's functional aspect of immune system that will protect these people. So those are still getting problematic, I think, in this picture.
I see.
There's one question from the participants, so I will now allow you to talk, João Paulo.
Sorry, I couldn't hear.
There was a
yes.
question.
Oh,
There
yes,
was-
question. Yes, I see it. Please share the best tips for. Oh, sorry, that's not the
I
question.
think the question from Joe was related to the previous presentation and was answered
Okay.
in the Q&A.
How
I-
did the SARS-CoV-2 vaccine question? That's the one.
Sorry, yeah.
Yeah, it's moving, so I couldn't see. How did the SARS-CoV-2 vaccine perform in comparison to other cohorts vaccine you studied on the high immunotropic groups? Was it better? Yes, of course. So SARS, if you look at the protection after SARS-CoV-2 vaccination, people respond very, very strongly, right? So we are not very worried about that. But there is more to that. That's what I was trying to explain. You still need functional immune system to protect yourself from the disease in the long term, because you lose your antibodies faster, or your functionality of your immune cells are still not as good. So the risk of infection is still increasing, although vaccine platform is giving sufficient protection after 28 days and 2 doses of vaccine.
Okay, thank you. There's another question. Can you please explain what the novel T-cell subset may consist of?
No, it's going to be submitted very soon, but so I cannot disclose what type of cell it is, but there is a lot of like not investigated heterogeneity in naive cells, and we know naive cell response is very important for vaccines and also for aging. So that's what I can say is a novel naive T cell subset.
And then final question. Have you seen patients change from the initial immunotype to another group after immunization?
Yeah, so this is a really good question. People, of course, move to a different configuration after vaccination. But what I can tell you, people, these high responder immunotypes, after vaccination, they are still closely related with each other before and after vaccination compared to everyone else. Because what you expect, what happens after vaccination, you move into new configuration. And if you have sufficient T cell response, B cell response, then you still look similar in that aspect, right? And what even happens, people move between the baselines, because we have influenza baseline, pneumococcal baseline, and you can see how much they move, and that even that is associated with immune entropy. So the rate of change of the immune system, the rate of aging, you may say, or like rate of increasing variation. Yeah.
Okay, thank you very much. So we are beyond the hour. So I want to thank all the speakers of today again for their amazing contribution. And we will share the information that Martin was sharing in the beginning of the webinar on our socials. You can find us on LinkedIn. The presentations were also recorded and will be available on YouTube. So you can find us on the social media channels. And thank you very much for your attention today.